All right, so welcome back. So we will be now having Professor Mohit Kumar Jolly of the Cancer Systems Biology Lab and Department of Bioengineering at IISc Bangalore. So Mohit, I've known for quite a while. In fact, if I'm not mistaken, Mohit, you also came here to give a job talk, right? Yeah. So yeah, unfortunately, we lost him to IISc, we have been talking to Mohit for a long time now about this connection between cancer and development. So it's a great pleasure to have him speak precisely on that topic. Design principles of complex cell-fed decision networks, examples in development and cancer. So this is going to be the part 1, and he's going to give the part 2 tomorrow.
All right. Thank you very much, Siddhabro, for Shakti, Fernando, and Mithun for having me here. It's a fantastic lineup of speakers, and I'm also impressed by the diversity of students, the disciplines you come from, the institutes you come from. It's great to have you all here, and I think this is a great time to, as Siddhabro was saying earlier, there is a critical mass of people around now who are thinking at this interface. Multiple collaborations going on between different experimental and theoretical groups. I'm sure you'll hear more about them. So please feel free to stop me anywhere wherever there are any questions. What I'll try to do in the 2 parts today and tomorrow, today we'll talk about how you start from simple gene regulatory networks and then slowly build up the complexity to see how the dynamics and the behavior changes, of course, while keeping the biological context in mind. And so this is more like a bottom-up approach. And tomorrow what we'll do is more a top-down approach, how you start from complex regulatory networks and see are there simple underlying principles that can be used there to understand the dynamics. Okay. So the theme of decision making has already been discussed in the first session and also during the introductory session. We're all here to try to better understand how cells take in diverse forms of information and then convert them into some discrete outputs. Those discrete outputs in this example being whether a cell will proliferate, whether a cell will move, whether a cell will differentiate, be able to survive, not go the apoptosis route, so on and so forth. Right? So cells are receiving these diverse signals, which of course vary both in space and time, as Shashi talked about the developmental examples. There is ample evidence now across different biological systems that if you take a set of cells which are all isogenic in nature, which means that they all originated from the same cell, same genetic background, still you see that they respond differently to the same signal being given. Right now, there are examples of this in drug resistance, in bacteria, in cancer. So cancer cells, which some of them do get killed by the drug, but some of them acquire a different phenotype which they did not have prior to the treatment. Right? So of course, once we understand what are the different outputs of the same stimuli, how do we control the proportion of cells taking those different outputs, and eventually can we manipulate the system so as to change that proportion to our desired outcomes. Right? So of course, this is an emergent property driven by interconnected complex networks, and we'll talk about some examples today. Many of you who come from biology background or may have read coming from physics background and have started reading more biology and cell biology kind of papers may have seen a schematic like this usually towards the end of the paper. Right? Now this is a great way to summarize, for instance, what the interpretation of those set of authors is in terms of the flow of information. So they may have done different perturbations which then somehow is stimulating this set of molecules that is triggering down the whole set of cascade, eventually leading to this output. Right? But now if I ask you, just looking at this diagram, let's say my environmental signal is oxygen, and I change percentage oxygen from 20% to 5%. And the question I ask is, tell me how many of cells will proliferate, how many cells will die, how many cells will differentiate. This diagram alone cannot give us that information because there is a complete lack of any quantitative details that is present here. Right? So what we do not have at all in this good first initial model, although not parameterized, not a mathematical model, but still a model in terms of a schematic, it is lacking time scales, it is lacking strength of regulation. So, is each of these phosphorylation reaction happening at the same rate? Are they happening in the same part of the cell? Are there spatial components involved? Is it direct? Is it indirect? What happens if you have two inputs coming on the same node? Is it an AND gate? Is it an OR gate? Is it a more complicated function than that? None of that information is present here. So in that case, what we tend to think of this system is a simple system where there's information flowing, and when we ignore all this complexity, that often leads to some major the assumptions that we are making here lead to some major misunderstandings in how exactly a system behaves. And when we start thinking about biomedical context, then those misunderstandings eventually lead to clinical impacts. I'll give you one example. In about 20 years ago, when people had identified that cancer cells have the ability to grow their own blood vessels, and the therapy that was in the clinical trials was called as anti-angiogenesis therapy, which means angiogenesis is the process of development of new blood vessels. And what this therapy was doing was stopping that process by blocking specific molecules. When this therapy was given to patients in the clinical trials, in some clinical trials, patients who received this drug started dying faster than patients who did not receive this drug. And it was only 10 years, 15 years later that it was identified that a major reason why this exact opposite response was seen in the clinic is because when you deprive cancer cells of oxygen, you are motivating them to move to other organs. You are triggering their what is called metastasis, that they would go on to different organs, usually using the blood vessel network which is already existing, right? So you're already seeing this complex system behavior here, this adaptive behavior here. The underlying assumption was they are dependent on oxygen. Let's cut off the oxygen supply, taken care of. But clearly there was another outcome which was worse than the outcome that they were already existing in, and that is what led to these consequences, right? So therefore there are these assumptions that we often make here can have these consequences, which again in the biomedical context we should be careful about. So what we'll do today is we'll start with some simple networks, a simple 2-node decision-making networks, 2 transcription factors for the sake of simplicity, interacting with each other in different manners. Then we extend it to 3-node, 4-node, and there are examples of this in T-cell differentiation that we have looked at, so we'll discuss that. And then we know that this is a huge simplification. Of course, these decisions are not taken by 2 or 3 or 4-node networks. Instead, they're embedded in much larger networks. So we also did simulations like this. We took these 2 and 3-node networks and then embedded them in much larger networks of varying density, varying sizes, and asking the question, is the dynamics unperturbed, or where exactly is the threshold that now you begin to see new dynamics which you were not seeing earlier. Okay. So these are the simplest 2-gene circuits. 2 molecules inhibiting each other, 2 molecules activating each other, and then 1 molecule activating the other molecule inhibiting. Of course, you can have it in the flipped manner, but that doesn't change the topology of the network in general, right? So this is what we would call a double negative feedback loop, because both the links are inhibitory in nature. This is double positive because both the links are activatory, and this is what is called as a negative positive feedback. So what we'll discuss today is how do these systems work, what is the dynamics of these systems, and why are these systems designed the way they are. So why in specific biological contexts you see system 1 instead of system 2? What are the distinctive properties which the biological system needs at that juncture to make a decision or to move ahead, right? So before we go into details, let's generally think about negative versus positive feedback loops, right? So both these are examples of positive feedback loops at the end of the day, because here the product of 2 negative interactions is a positive interaction. So A inhibits B and B inhibits A. So if A is high, that will suppress B, and because B is not present, A will continue to be high. So effectively it's a positive feedback loop, and so is this case. But this is a negative feedback loop. A is increasing B, but B is repressing A, right? So that's the main difference. So these both are 2 forms of positive feedback loop, and this is a negative feedback loop. And negative and positive feedback loops have very different dynamics, and we'll see some examples in a minute. So let's look at the system in our body. That it's a physiological example. Let's zoom out. Let's not look at a cell. Let's look at an organ or a system of organs, and see how is our body temperature controlled. So we all know that our body temperature is maintained in a given range, and if it goes too it crosses the range on either side. If it goes too high or too low, there are mechanisms to bring it back to normal. And when these mechanisms get disrupted due to various different scenarios, then is when you have, hyperthermia or hypothermia kind of examples. So here, let's say the body temperature increases. When the body temperature increases, the signal goes to the thermostat, and that activates your sweat glands. You begin to sweat, and that leads to cooling of the temperature. And also you have dilation of the skin blood vessels, and heat also is radiated from the surface of the skin. So this is a restoring force that brings the temperature down. Now let's look at the other direction. When your body temperature goes low due to certain reasons, then what you begin to have is shivering, and shivering generates more heat. And in this case, your skin blood vessels constrict, therefore preventing the heat loss from the surface. So this is also a restoring force which brings the body temperature up. So when it goes high, the system brings it to low. When it goes low, the system brings it to high, right? Let's look at another example, blood pressure. So blood volume is regulated by this molecule called aldosterone. Don't go into the details of this. The whole point is that, again, if the blood pressure increases, there are feedback loops to bring it back. And when the blood pressure decreases, there are feedback loops to bring it up. Third example, glucose levels. So again, there's a homeostatic range of blood glucose levels. When it increases, what begins to happen is that body begins to store glucose now as glycogen, and that is why you see it comes back. And when it goes low, then whatever the stored glucagon was there begins to be converted to glucose. It is broken down, and therefore you see it goes up, right? So all these three examples that you saw, body temperature, glucose level, heart pressure, these are all, as you may know, also vital signs of the body, right? When you go to a doctor, you're taking your blood pressure, your temperature is measured, right? And that is a clear signal that it should be maintained in a given range. So anyone would like to comment on why are all these vital signs mostly regulated by negative feedback loops? Is there a reason? Why is this not regulated by positive feedback loops? Why are there restoring mechanisms in the body rather than if it goes high, there should be a system which makes it higher, or if it goes low, there is a system which makes it lower? Please. That will cause exponential growth of the perturbation, you mean? Right, absolutely, right. So what you need is, the whole concept of homeostasis is, what does homeostasis means? That you maintain it in a specific range, right? So it's either it's oscillating potentially, either sustained oscillations or decaying oscillations. That's a separate discussion. But nonetheless, it is maintained in a specific region with not too high amplitude, and that is when the body functions the way it is supposed to function, right? So there are reasons why all these vital signs are regulated by negative feedback loops, right? The way you can, if you want to write down a mathematical model for this, you can keep changing the variables, but the central structure of the network will not change, because that is what the body needs in this case, right? Now, these are all at organ level, right? Now, let's think about tissue level or cellular level negative feedback loops. If they exist, what is their role? And what happens in the case of cancer? So in the case of cancer, as many of you may know, our normal, the non-cancerous cells, they have something what is called as a Hayflick limit, right? So there's a limit, the number of times a cell can divide, which is maintained by the whole telomere pathway. But in cancerous cells, that limit is, through different mutations, not obeyed, and therefore cancer cells can continue to divide many more generations as compared to a normal cell, right? So there are these negative feedback loops even within a normal cell. This is one of the most well-studied one. There are multiple mathematical models for this and the more extended models of this out there, called P53, which is also the most commonly mutated gene in cancer. P53 activates transcriptionally this molecule called MDM2, and MDM2 actually binds and degrades P53, right? So this is, one of them is transcriptional, the other is a post-transcriptional scenario here. But again, let's not go into those details. Let's just think about mechanisms operating at a similar time scale, and let's think about the structure of these cases, right? So what happens when mutations in the P53 mutated version, most of them, this loop gets broken. The MDM2 is not able to degrade them, and therefore the homeostatic limit is thrown off in various cases, right? Let's look at another example. So this is within a cell. Also, there is a phenomena called as contact inhibition of proliferation, which basically means that when a cell is has to take a decision whether to divide or not, it is through various different molecules, one of which is mentioned here. It senses how many cells are present in its environment and calculates that, and based on that, a decision is taken whether to divide or not divide, which is what is called as, if there is already a high density of cells here, it chooses not to divide, and that is what is called as contact inhibition of proliferation. What happens in cancer? This molecule is lost or is mutated. Again, yet another example of negative feedback loop being broken. Please. Is the? Yes, we are talking of mean behaviors here. We are not getting into stochastic cell-to-cell variability concepts yet. Those are important questions, but we'll get to that later. Yes, absolutely. Okay. So negative feedback loops play a crucial role in homeostasis, and some examples of which get broken can lead to disruptions in organ architecture, function, eventually leading to cancer. So then the next question comes, is a positive feedback loop seen anywhere in physiology? So what would a positive feedback loop do? As was already mentioned, it would amplify the perturbation, right? So when you have amplification of perturbation, you could have more divergent responses rather than more convergent responses, right? It cannot be sustained for long, okay? Anyone, please? So, sorry, can you give the physiological example?
For example, even in case of negative feedback loop, for example, when we want to say release some hormone for the negative feedback, the cells that are releasing the hormone,
yes,
inside the cells there would be a positive feedback loop to give the signal itself, the amplifying molecules that will give signal to other molecules so that they can have increased simulation of
but
that.
What will stop that eventually? Something has to stop it, right?
When the signals come that the situation is maybe the
so you're saying a positive, if I understood you correctly, feedback loop is triggered based on the presence of a signal,
yes,
and when you remove the signal, this feedback loop goes away. Okay, that's a possibility. Any physiological examples of this? Okay.
So building on what he said, it seems like if there's a virus which is attacking, then if the immune system discovers it and the virus fights back, probably the immune system would send more forces to fight.
Okay, okay. That's close to one example. I'll get to that in a minute. Any other examples? Please. Okay. So how is that amplifying, and why is that amplification necessary? Because we're talking of two things, how and why.
Okay, the way of amplification is the hedgehog signal goes to engraved cells and stimulates them to express hedgehog and engraved.
Okay.
And the hedgehog goes to the wind cells and stimulates them to express wind.
Okay. So what happens after that? So some signal is being transduced in a layer of cells, right? But why is it necessary there?
It maintains high levels of hedgehog and engraved to maintain a gradient on both sides.
Why is that gradient important?
The gradient helps mark the boundaries of segment spacing.
Okay, so you're talking about a spatial segregation in this case. Okay, okay. Please.
In case of immune cells, when T cell binds to a MHC, MHC2 molecules basically, it releases cytokines, and then it again helps in release of more cytokines by the end proliferation of the cells,
Okay.
which is a positive feedback loops which helps in proliferation of T cells, which basically ends up making the immune response more robust and more potent.
Okay, okay. I think so all the answers given have different components which will fit in the examples that I'll present. But before I go there, so as we discussed that, right, positive feedback loops amplify a response, right? So let's say you have a signal coming in, and as was pointed out, that signal can trigger a positive feedback loop which could amplify the perturbation and take some cells to a completely different state as compared to where they were initially, right? Is that what was said? And some cells which somehow for whatever reason either do not respond to the signal or do not get enough signal will not be able to go to the other state, right? So you'll have, in many cases you will have a possibility of two distinct states which are very different functionally, physiologically, molecularly, and those two states could not have emerged if this were a negative feedback loop. Is that okay? Okay. So now let's talk about this example. So this is the example of childbirth. So when the head of the baby is stretching the mother's uterus, that is triggering a whole bunch of responses involving the nervous system, of course, that is making the contractions of the uterus even more amplified, even more powerful, right? And it is these contractions which is then further triggering, as was rightly pointed out in one of the answers, the release of the same molecule, oxytocin, and what happens is that this cycle continues, right? So what is the signal? The signal is the head of the baby pushing against the wall of the cervix, and that is triggering a whole set of response, eventually leading to higher and higher contractions and higher oxytocin levels, correct? Okay, so what happens as a consequence of that? What is the final outcome? Childbirth. Okay, but that's only one state. Where is the second state? And what happens when the child is born? Then this signal disappears and the system restores. What is the second state? There is no childbirth. It just stays in there, right? If a 3-month-old baby's head is pushing, the signal, would that be strong enough to trigger this positive feedback loop and eventually get it out? Probably not, all right? So we do have cases of preterm birth and so on, but largely speaking, it is the strength of the signal which is determining whether the system would switch to a different state or not. So the system has the possibility of existing in two states, baby in, baby out, but what is deciding the switching between those two states is the strength of the signal, which is then going back to the size of the child, the time of gestation period, etc., right? So that's one example. Do you need this amplification in this case? What if this were a negative feedback loop? There will be no childbirth because the system will bring it back. So you need a positive feedback loop in this case to have that discrete event being recorded in time and at the right time. Okay. Let's take one other example which was also mentioned in some form, blood clotting, right? So when you have a wound in your skin, or a break in the blood vessel wall, that triggers a whole set of responses such that platelets come in there and try to make a clot, and those platelets are also secreting cytokines to get more platelets there, and that is eventually leading to the clot being formed. Now we have to be careful here because unwanted clots is a cause of heart attacks. You don't want your blood to be clotted randomly, right? So there has to be some signal which cuts this off, which cuts off this positive feedback loop. In the childbirth case, what was that happening? The child being born, signal is gone. So here also there are other, it's a coupled positive negative feedback loop. So the negative feedback loop gets triggered only when the clot is formed and brings it back. So we are talking about very, we are thinking as if these are very simple cases, which they are in some cases, but they're also integrated and coupled to each other. So positive feedback loops and negative feedback loops don't exist in isolation. They are coupled to each other, they can have different time scales, etc. We are talking of a gross oversimplification as a first exercise in general. Okay. Any questions in this so far? Negative feedback loops, restoring, positive feedback loops, amplifying, possibly leading to two states, also called as the phenomena of bistability or multistability, which we'll go in more details of. Is it okay? Please. Yes, in this case, yes.
In that case, so for example, when you said when say the child is in the gestation period, 3 months or so, it's the strength of the signal that won't allow it for the positive feedback.
To be
So
triggered,
my
yes.
doubt is, how do we know it's the strength of the signal, or there may be that negative feedback, say, is there when the child is, say, some 8 months in the womb, and when it comes to 9 months, then the cycle switches to a positive feedback. How do we know that's not the case?
No, I'm not saying that's not the case. That's absolutely right. That is also a possibility, that somehow there is a break which is, you know, maintaining, which is not letting the positive feedback loop being triggered. So what you're saying is that at some duration, it switches from a negative to a positive feedback loop, right? Or in other words, you know, you remove your foot from the brake and you press on the acceleration, right? So again, there is some timer. So whether you call it timer, whether you call it strength of the signal, those are, you know, different semantics to define a similar dynamical behavior. Is that acceptable? Please.
So how do time scales play a role in these feedback loops? For example, your example of a 3-month child pushing. So even like if it's a positive feedback loop, that like it'll keep accumulating over time and oxytocin will keep releasing. But is like how does a time scale play a role here? Like over how long does this oxytocin have to be released for the effect to be like, you know, noticeable or something?
Right. So that's an important question, right? That where are these time scales coming in, right? What are the different events happening here? Release, sending the signal to the hypothalamus. That's a very quick thing, right? Reflex actions, we all operate in that sense. Then there is, you know, oxytocin being released. Was oxytocin already there in sufficient quantity, or do new molecules have to be generated? That leading to traveling in the blood to the muscles of the uterus. This is seconds to minutes time scale. It's not more than that. So it's once it's triggered, it's a fast response, and then eventually leads to these two outcomes. Of course, yes.
External resources that the body has.
Absolutely.
So that will always put a cutoff to any positive feedback loop.
Yes, yes. So there has to be, otherwise the system will just not stop. So there has to be some discrete event which cuts off that positive feedback loop. Okay. All right, so let's come back to these examples again. Well, let's dig a little bit deeper. Now that we know how negative and positive feedback loops function, let's see if those same concepts apply when you look at, you know, our two-dimensional dynamical system at the end of the day, right? So now, just to bring everyone to the same page, this is a central dogma of molecular biology. Gene gets converted to mRNA, which gets converted to protein, which leads to these different cell processes. And some of these proteins have the potential to alter the rate of transcription of themselves or of other genes, which is what is called as transcription factors, right? So what do transcription factors do? So let's say this A is a transcription factor, and you know, this transcription reaction in absence of A was happening at some fixed rate, and in presence of A, that rate gets amplified 10 times. I'm just making up numbers, this is just an example, okay? Or what could also happen is that A could reduce that rate of production of C to 10 times, right? So if it increases, it's called a transcriptional activator. If it decreases, it's called a transcriptional repressor, right? Which could be, again, you can draw parallels with the brake or accelerator in a car. Now let's say that A is an activator of B. Okay, so how many of you think that if I were to plot the levels of B as a function of the levels of A, it would be a straight line that passes from 0, 0. Is this correct? If A activates B, is this the right mathematical form or expression to denote that? Yes, please. It should be a step function. Okay.
If it binds, then
but
in most cases.
Will the step come at A equals to 0?
I don't. At 0, it will be 0, and just after that, it should become.
Just after that. Okay. Okay.
Basically, I think threshold. After a threshold of A,
so then
it goes to.
Why would that threshold exist?
to be able to make sure that there is no false positive. Basically, if A comes after a certain threshold, then only B gets activated before, so that there can be basically you can determine whether the signal is robust and if the action is really required.
Right. So you're answering it from a design principle perspective, that why would have system chosen this? Okay. Can you try to go at a molecular level? Why would there be a threshold? Please. So sorry, I didn't get you. Can you please repeat?
Molecularly, say if one molecule gets released, then even one molecule will start accumulating a positive feedback cycle.
Okay.
And then it won't stop. If there is a
Again, that's a design principle answer. I'm asking a molecular answer. So, okay, think of any transcription factor. Can all of you name some transcription factors here? P53, okay. P53 is a transcription factor. How many targets it has? 5, 50, 500, 5,000, 50,000. Any guesses? Just 1 second. Any guesses?
The level of E can be because of some other factors. So if there is A based on some positive feedback of other targets of the, for example, P53, then it can also affect the activation of B, for example. So
Okay.
only if the extent of A is such a way that it's been produced by effect of B, then it should have a threshold, because there might be a minimal level of A always in a cell. So only if the extent of A is so high that then it can have a positive feedback.
Right. So that's getting closer to a molecular answer, right? Please. Okay.
Because molecularly, let's say A is a system, B is a system, both of them have C as a complementary part,
Fine.
but A requires 2 molecules of C, whereas B requires 3 molecules of C. Now that is what threshold does.
Okay.
So yeah.
So that's, again, that's the design principle answer. So, okay, what is happening here? A is a transcription factor. Where is B? B is on the DNA. And transcription factor is going and binding to the DNA, right? Is the DNA just sitting idle waiting for A to come and bind? There are various other things happening, right? And is the binding sufficiently long enough that it can recruit RNA polymerase and transcription can happen? Please answer.
What are the fundamental limits on how many transcription factors we can have?
Why would we have a threshold? Yes, that's molecular reason for a threshold, right? So there are all these processes that have to happen, each of which is a stochastic reaction, each of which may or may not happen in the time step, that time interval that you are looking at, and therefore you would need threshold levels of A to ensure that those events all happen at an ensemble level, and you could trigger the increase in production rate of B. RNA polymerase can come, it can stay there, it can increase the rate of production in presence of A, right? So that's one thing. So you would need a threshold. What about this part? Will there be a saturation or no saturation? Will there be a saturation? Why? Please. Sorry. Right. And there are only so many A molecules. Both of them, right? So there will be a saturation. So this graph is wrong in 3 manners. A, it will not pass from 0, 0. Then it will not be unbounded, and it will not be linear. Is that correct? Right? So basically, in general, what people have observed is that you get a graph something like this, which to many of you may remind of bacterial growth curves in some sense, right? So there is the so-called lag phase, then there is a growth phase, and then it again sort of saturated. And the slope of this curve determines how strongly A affects B. So if if A is affecting B very strongly, then you will begin to see the function getting more and more steep and basically, tending close to a step function eventually. Okay. Now, what happens if A represses B? How will the graph look like then? So again, there will be some threshold, right? So you will start from high levels of B, right? Because the A was low, so B's steady state levels were high. And then in presence of A, you will begin to see a decrease, and then again it will saturate. Is that correct? So some function like this is what is often sort of, reported in the literature. Okay. Now, if I were to ask you, can you write down a mathematical expression that fits this kind of curve? What would that be? Any mathematical, please. No. It is that's a plotting typo. Sorry? 1 minus e to the minus x. Okay. Anyone else? You can tell for either of them, either the top one or the bottom one. We can easily get the other one. That's not an issue. You can just normalize it to 0 to 1 and then subtract 1. Okay. How about something like x square over 1 plus x square? Would this fit the top? Well, what happens at very low levels of x? What is the value of this function? 0. At very high levels of x, what is the value of the function? 1, right? And then in between you would see a sigmoidal increase. Why sigmoidal? Because there's this clear non-linearity which is present here, right? So this is what is called as Hill's function. It was identified by someone called Hill, and there are again some justification given for which I'll just go into in a minute. Some justification which is often given for why these kinds of functions may have a molecular basis, right? So that is the following. So let's say you have, let's go to the elementary reactions. So there's a transcription factor which is binding to DNA. There's some forward rate, there's some backward rate. Let's just replace with A and B. I'm sorry, these are the same names, don't get confused. But nonetheless, AB is the bound molecule, and you have transcription factor and DNA here. Now, if you were to write differential equations for this, rate of change of AB as a function of time is just the forward reaction Kf times A times B minus the backward reaction Ki times AB, right? And what about dA dt and dB dt? It is just the minus of that, because minus Kf times AB plus Ki times AB, right? Now, we make an assumption. What is the assumption? That the DNA and protein binding, or the DNA transcription factor binding, happens much faster as compared to actual transcription. Now, if you make that assumption, you say the rate of change is 0, you equate those two terms, you get some term K equivalent, which is basically AB times A, AB divided by A by B. Okay, so what you have here, you have basically the fraction of DNA that is bound. You're trying to get the fraction of DNA that is bound, right? So what would be the fraction? That fraction would be bound DNA divided by unbound plus bound DNA. And because you have AB in terms of B, which you just calculated on the last slide, you could replace that, you could do the arithmetic, and you get a term which is like x over some constant plus x. You can set the value of that constant as 1 just for simplicity, right? But here we wrote down something x square over 1 plus x square. So where does that come into picture? So now instead of one transcription factor molecule, if you have multiple of those molecules, so you get, you have the first reaction TF plus DNA binds to TF DNA, and then this one will bind to yet another molecule of TF and give rise to 2TF DNA, 3TF DNA, etc., etc. Now, if all these reactions are fast enough, basically, what you're saying is that as soon as this gets formed, there's a high tendency to bind to another transcription factor to find this complex. You could eventually get, so what you would have here is some something like this. But again, if you're ignoring this, which basically means that the single bound complex does not exist because the rate of forward reaction is so high that it eventually goes to that, right? So this is an assumption. There are again some molecular justifications for that which is available, that binding of one molecule can somehow change the structure in such a way that binding of the other transcription factor becomes much easier, and or the transcription factor itself forms a dimer and then goes and bind there. So there are multiple of those explanations which are available, and again in some systems these have been seen in general. Please.
In every state are similar.
Yes, for sake of simplicity we are considering that.
Yeah.
Yes. Okay. So again, please.
In the denominator?
So if you don't even let the molecule exist here, as soon as it reaches here, the other reaction is, so here again basically the Kf for this molecule is much larger than Kr. You're pushing the system constantly in this case.
Okay. When the forward reaction is,
yeah,
is more dominating.
Yes. So again, this is just a graph showing that as you increase the value of n being the exponent here, you begin to see more like a step function response if you increase it all the way to 50, right? So for x square, you see a sigmoidal, but eventually as you increase the nonlinearity, you'll begin to see a step function like this. Okay. So now this circuit, these two molecules inhibiting each other, this was actually constructed synthetically. It was, and put in bacterial cells, E. coli, almost 25 years ago. And this is where synthetic biology kick-started. Okay? So what they did is they made this structure, which is basically the same as what I just wrote here, and it's just written in a more complicated form. And they wrote down this set of reactions. So again, U and V are A and B. Alpha is some production rate. And again, you have this nonlinearity here. And so this is the rate of production. So what this says is that the rate of change of U is some constant multiplied by, because it's an inhibitory function. This is the form we wrote down for an activatory function. But if you do 1 minus it, you get 1 over 1 plus x square, which is what is written there. And then you have some constant degradation rate. Okay? So again, for the sake of simplicity, they have taken very few variables. Alpha 1, alpha 2 being the production rates, and beta and gamma is the nonlinearity that they have taken. Degradation rates have assumed to be of unit value in this case. Okay. Now, in this case, if you were to look at these reactions and say beta and gamma are both 1. So you have du dt equals to alpha 1 over 1 plus v minus u, and dv dt equals to alpha 2 over 1 plus u minus v. Okay. Beta and gamma are both 1. Would that lead to two discrete outputs such as u high, v low, and v high, u low? Right? If two molecules are inhibiting each other, you can get these two extreme outputs, a high, b low, or a low, b high. But would that happen for beta and gamma equals to 1? How will you figure that out? So if you want to solve these equations for steady state, so what would be you put du dt equals to 0, what do you get? You get alpha 1 over 1 plus v is equals to u, and from the second one you get alpha 2 over 1 plus u is equals to v. Right? Now you plug in the value of u here, you plug in the value of u here, so what do you have? You have alpha 1 over 1 plus alpha 1 over 1 plus v is equals to v. Right? You will get a quadratic in v. How many real distinct solutions can a quadratic have? A maximum of 2. Okay. And for a bistable system, how many steady states you need? 2, 3, 4, 3. Why 3? There has to be an unstable state in between, right? Between every two stable states, there has to be an unstable state. So clearly, if beta and gamma are both 1, you will not get bistability. You will not have this mutual exclusion being drawn. U and V will coexist at some values. So clearly, if you have a circuit, that doesn't mean that every single value will give you those outputs. So this is the analysis that they had done. They had shown that, as you keep increasing the value of beta and gamma, there are larger and larger regions in this parameter region of, in this phase plane of alpha 1 and alpha 2, where you get bistability. Because the higher the nonlinearity of the system, the more the likelihood that you would get, these two distinct solutions eventually in that entire parametric space. So they constructed the system. What happened after that? Now here, again, let's not go into details. Just think about this as signal 1, this as signal 2. What is signal 1 doing? It is activating A. So it is trying to push the system to A high, B low state. And what is the other signal doing? It is trying to push the system the exact opposite way. Okay? So you start from, and these are just different bacterial strains, and these are control cases where this system was not working because they did not put in the feedback loop, they put in only one side of it. Okay? So here you can see the system as a function of time. You could push the system to this state where, again, they're measuring one of the molecules, let's say A. So A is high. When you remove the signal, because this is a stable state, the system will stay there. It stays in that A high state. And you need another input to push the system back to an A low state. And again, when you remove the signal, the system stays there. Right? So this is clearly a property of bistability, that you see that the system could stay in a state, but also under perturbations, you can switch states. So you could have two distinct states in which you could toggle, and that's why this is called a toggle switch. You could push the system back and forth between two distinct states. Right? Now, what are the consequences of this kind of behavior? Right? So let's say if you were given a Western blot, which is a population average behavior of a layer of of a population of cells, and you increase the duration or concentration of stimulus. And what you see is that the protein levels increase. But now if I ask you, is that happening because in every single cell, the proteins are increasing, the protein concentration is increasing slowly? Or is it that some cells are switching to a high protein state and the others are not? And what is increasing as a function of duration or concentration is the percentage of cells which have switched. Right? So here you see whatever, 10, 20 shades of gray in between, and here you only see black and white, and what is controlling is the percentage of cells which are in black state or white state. Right? So this can have very different responses. This is what would be allowed by a bistable system, because you have two discrete states. This is what is a monostable state. You are just continuously increasing the rate of production, and that is why what you do not see is this splitting of population structure and then again merging of them. So when they did the experiments, they found exactly this, that at earlier time step, all cells are in state 1. This is a histogram of cells, so flow cytometry. At some other time point, say 4 hours, when at an what you see is that this peak splits. Now you have a set of cells here. Within 5 hours, you see a large proportion of them switch, and within 6 hours, everyone has switched. So what you not see is a constant just moving of this distribution. What you see is splitting and then merging of this, which is very, very typical of any bistable system. Right? Now, what are the consequences of this? Why does it matter in this case? So again, I'll present one example. This is in the context of bacteria. So when this has been seen in many, many bacteria by now, that you take a bacterial population and you increase the dose or time of the antibiotic that you are giving, and you hope that all cells eventually die. No matter what antibiotic you take, no matter how much time you take, some cells are always left behind. And when you regrow the bacteria, the entire colony, using those some cells which are left behind, and redo this experiment, you again see the same behavior. and you can repeat it over generations and generations, and the dynamics does not change. Now, if these two responses were happening because of genetic changes, that there are irreversible changes in the DNA which is causing this, then when you regrew the bacteria from that small population, what you would have seen is a curve like this, where it just does not respond to the same antibiotic, because those cells are privileged enough for whatever reason that they are not responding to that drug. So the fact that you see the same dynamics repeated again and again clearly argues that it is not genetic in nature, it is instead phenotypic in nature, that despite having the same genetic background, you still see two discrete outputs. And this is what has been shown, that indeed bacterial cells have, they are in a bistable system and they can switch back and forth, and that is why you never see populations disappearing to zero. You always see some populations are left behind. And of course, the entire field is trying to work towards how do you get cells out of that second state back to a sensitive state and then eventually kill them. Okay. So this dynamics, which we talked about in a simple example, has implications in major biomedical health scenarios as well. Okay. Let's look at the other system which was constructed. So this is now 3 molecule. A inhibits B, inhibits C, and C inhibits A. So it's again like a negative feedback loop. It's not a two-dimensional system, but the dynamics are very similar. This was the second paper that appeared in that journal Nature issue when the toggle switch paper did. And again, they did show that as a function of time, you could see these oscillations in a population of cells which had this construct, and the cell population which did not have this construct, or they did not show these kinds of synchronized oscillations, right? Now, of course, you can see that the y-axis value is increasing. That's happening due to multiple reasons, because the number of cells is also growing, etc. But clearly, this dynamics was also shown 25 years ago. So, these two systems have been constructed and have been put in cells, and they behave the way dynamical systems theory would predict that these two behave, right? But since then, even the simplest extensions of that, which we will just look into, have not yet been constructed. Okay, so we will think about it, why possibly that is the case, right? So let's go back to toggle switch, and if you look at development, right? So you have the single cell zygote here that could take any of the two branches, any of the two cell fates, and then you can continue to see how different cell fates emerge. And in most cases, what you see in this map is that there is a bifurcation happening at this, and the same progenitor cell is able to give rise to two distinct populations. At each of these branch, what you see are two transcription factors which are inhibiting each other. The name of the transcription factor can be different depending on which branch you are looking at, but eventually the two states that you see, A much greater than B or B much greater than A, are the two sister fates that you eventually get. It's like, if a progenitor cell has to decide whether to go path 1 or path 2, and those cells have to stay there, you want the other path to be switched off, and switched off reasonably strongly, right? You don't wake up next morning and your heart has become a lung, or your lung has become a heart, right? So you want these mutual exclusive kind of gene expression patterns, right? And those patterns are mediated through this feedback loop. So again, going back to the why question, right? Why would we need this? We need this because we want diversification of fates happening. You start from a single genome, but you want 200 outputs out of it. How does that happen? Because molecules are interacting with each other, giving rise to multi-stable dynamics, such that you could eventually have cells, of course, coordinated in space and time, which we are not talking about here, but you want those fates to be chosen in a reasonably non-spurious manner, right? Now, when there's an injury, a major injury, so there are these so-called adult stem cells that many organs of our bodies have, which have the capability to then act as these progenitor cells and give rise to all the other cell types. So there is backup being maintained here, but that backup does not come at the cost of interfering with cellular decision making, which is needed during development. Okay? So we sort of started thinking more carefully about this, that why do most branches have 2 or maximum 3, why do most junctures have 2 or maximum 3 branches coming out? What is preventing the same progenitor cell to give rise to more than 3? Okay, so that is the question we sort of started with. This is just more explanation of toggle switch. We have already sort of discussed that. You could write down these set of equations similar to what I wrote down here. You could just add more variables to increase your parameter space. So there's a rate of production, there's a rate of regulation, and then there's a rate of degradation. And again, these are the null clines, how A changes with B, how B changes with A. This is the unstable state, and these are the 2 stable states, A high, B low, and A low, B high. Okay. This we've already talked about, that it's not always bistable. Now, if this was a double positive feedback loop, right, so this equations would change. Instead of B naught, you would have B in the numerator because it's 1 minus this, and these null clines would also change. So if both of them activate each other, you would also have 2 states, but those 2 states would be both low or both high. But if you have both inhibiting each other, you get these 2. So, right? So toggle switch is not the only bistable scenario. You do have other cases of bistability, but in the cases of decision making, you want toggle switch because it allows for stable self-rate decision making, which sort of progresses in time. Okay. So again, many of you may have heard of this, or I'm sure you'll hear about this a whole lot in this workshop. This is what is called as Waddington landscape, which was drawn by Waddington in 1940s at that time, when we did not know what DNA is, we did not know what genes are. But the general idea here was that you have a cell which has the capability to give rise to multiple cell states, and as time progresses, the number of options that it could give rise to continue to decrease as a function of time, and eventually a cell ends up in one of these ridges or one of these valleys. So here a ball is representing a cell fate, and this landscape here is representing the cell fate trajectories as they progress in time in general, right? So a ball rolling down and ending up in one of these landscape means that a so-called pluripotent cell has chosen one of those paths of differentiation. Okay, but now as we know that this is not a one-directional flow, cells could switch between these ridges, between these valleys. Cells could go up this landscape, which is what got the Nobel Prize, induced pluripotent stem cells that many of you may have heard about, which is making major strides in regenerative medicine at the moment. So cells can change their identity in a reversible manner. It's no longer irreversible as was initially thought of, but again, they do this in a very, very controlled manner, right? So this got the Nobel Prize 2012. And I think it was published in 2007 or 8, this initial paper. Anyone would like to take a guess of what is the, so let's say if I were to take one of these so-called differentiated states, in this case let's take fibroblasts, and you take fibroblasts and you give whatever perturbation you have to give to it and try to make it a stem cell. What is the success rate? Any guesses, please? It's less than 0.1%. It's that bad. Okay, so clearly, again, let's go to the why question, right? Our body has been designed in a way that you can push it, but you would need really, really hard to, push it back to that extent in some cases. So clearly, this is a very open area of research. If you want to make, just one second, so fibroblast to neurons, fibroblast to kidney cells, etc., etc. These are all open questions in the field. What exactly are those networks? What is the dynamics of those networks? How can you, eventually convert this into what is called regenerative medicine? Please, all the concepts of dynamical systems are directly applied there. Yes, please.
So, for one, the success rate is very low, and once a cell has been converted to, let's say, a pluripotent state, is it true pluripotent as it was in the, let's say, embryonic stage, or is it some sort of partial pluripotency?
It's more like partial. That's why they used a different terminology for it. They didn't call it embryonic stem cells. They called induced pluripotent stem cells.
And so once, so let's say we take some fibroblast cells,
right.
we induce pluripotency and convert it to some, let's say, muscle cell or something.
Sure.
How different is this muscle compared to like a normally differentiated muscle cell? Are they physiologically the same?
So that's a very good question. I think some functional properties, so the first check is the molecular expression, right? Whether whatever state you have gotten is a molecular mirror of what a muscle cell should be. So you choose your favorite markers and you show that yes, it is similar enough. The next check, as you rightly pointed out, is the functional check. And their efficiency goes down even further in the sense that cells may be showing the marker but not functionally active. So the 0.1 I think includes both of these steps in that sense. I don't know the exact distribution of where it fails.
Thank you.
Please. In most of the examples, no. In fact, I'm going to talk about examples where it's not toggle switch. Just a minute.
Success rate for transdifferentiation.
Sorry?
What's the success rate for transdifferentiation?
That's a good question. That depends on which pair you're talking about. And again, I think again that the whole concept of transdifferentiation is not extremely well defined. The point is that it does not go all the way to being a stem cell, but it goes maybe partially up somewhere and then goes there. Now, how do you set that boundary? Right? How many steps you have to go back? Would you call it transdifferentiation versus dedifferentiation? But the success rate is again not more than 5% to the best of my knowledge. I may be wrong. Maybe there are more developments. I've not followed the literature. Okay. So I'll present two examples, one today and one more in detail tomorrow. So we did look at T cell differentiation, which actually a T cell could give rise to at least 4 or 5 different states. For the same T cell, you give it different cytokines, you get these different states. People do these experiments on a regular basis. This is CD4 positive T cells. Okay? So one of the cases where we found enough literature to construct this network was these 3 molecules which inhibit each other. So instead of a toggle switch, now you have a toggle triad. Okay? 3 molecules. These are all transcription factors. TBET, again, let's not worry about the names, let's call them A, B, C, but let me just introduce them once. So TBET drives this TH1 phenotype, GATA-T drives TH2 phenotypes, ROR gamma T drives TH3 phenotype, TH17 phenotype. Why they are called 1, 2, and 17 is based on the interleukins that they secrete. Interleukin 1, interleukin 2, and interleukin 17. Does TH3 exist? I don't know. It may exist and has not yet been found out. Does that mean that 16 exist in between? I don't know. Maybe there are more and more papers coming out identifying. Now there is TH22, TH23, etc., etc. Okay. Nonetheless, let's start with this simple case. 3 molecules inhibiting each other, and what we know experimentally is that these 3 states exist: A high, B low, C low; A low, B high, C low; A low, B low, C high. These 3 states exist. It has been shown. Okay. So the question we were asking is, can this dynamics explain these states? Or in other words, what are the states that you get when you simulate the dynamics of this network? Now, what we do not have is the exact parameter value of these interactions. So what we did here is we wrote down very similar ODEs that what you have been seeing so far, and simulated the dynamics of this over a large range of these parameter sets which were chosen from a biologically relevant range. So we don't know the exact half-life of a protein, but we know the range of half-lives of proteins in mammalian cells. So you could choose that range and say that I'm going to sample my range of half-lives from this. Similarly, you can do this for production rates and so on. So when we do that, so what you have here, what are the inputs? The inputs is the topology of the network. Topology means just the interactions that you just saw. You could define a parametric space, and what you get is an ensemble of kinetic models, and for each parameter set you can start from multiple initial conditions because you could be in a potentially multi-stable region. Okay. When you do that, what do you get? What we see is the following. when you take, we did this for 10,000 parameter sets, and for each parameter set we started from 100 initial conditions. Okay, so this is 10,000 cross 100 simulations done. Each row is a solution of the system. So each row is a virtual cell which has slightly different kinetic parameters than the other row. And then you take all the parameters, all the solutions, and then you basically normalize it on a scale of z-score, so -3 to +3. Okay? Now what you see here, if you plot this in silico heat map, is that there is this whole bunch of solutions here, red is high and blue is low, where A is high, B and C are low. This whole bunch of solution here where C is high, A and B are low, and this bunch of solutions where B is high and A and C are low. So you get these phenotypes as a consequence of these cases. And you also see predominantly this system is monostable. 50% of the parameter sets lead to only one state, but you do see some instances of bistability and tristability as well. Let's quantify further. How many solutions lead to these states where one is high, so the capital indicates, the capital letter indicates that that gene is high. So either C is high or B is high or A is high. This is the most dominant behavior. But in addition to that, not too far behind, is two molecules being high simultaneously. And what you do not see is all low or all high, right? So you only have 3 variables, and if you classify them into high and low, you get only 8 possible states: all low, all high, single positive, and double positive. So all the 3 single positives is still the highest, followed by double positive, followed by this triple positive or triple negative. So you get these states, but in addition to this, you get these hybrid states as well, right? So you could switch from state B to state A by going through this hybrid state, and similarly all these other possibilities exist. We showed that these states could coexist, so you look at the most frequent tristable states. It indeed is the combination of those 3 states. Most frequent bistable states is the combination of any of those 2 states taken together, etc., etc. And then the next question we asked was, how unique are these properties to this structure? So let's say if I were to take this structure, A inhibiting B, and now if I convert one of these links to activation, do the properties change, right? So you could do these perturbations. There are, I think, 13 or 14 such non-biological hypothetical networks you could generate. And what are the properties of a toggle triad? The properties are that all high and all low do not exist, or very low frequency. All 3 single positive exist at a similar range. All 3 double positive exist at a similar range, and single positive is more than double positive. Those are the 4 properties that we saw in that graph. Okay. Now let's look at these networks. So these are the other networks where we have replaced one or more with activations. These networks, which you just saw change their color, do not follow this property. These networks do not follow this property. These networks do not follow this property, and these networks do not follow this property. So there is not even a single network which follows all the 4 properties. So clearly the dynamics of that, or the graph that you saw, that all low and all high are very low, there are single positive states and then there are double positive states, and single positive is more than double positive by approximately 50%. None of the other 3 node networks follow this property, suggesting that these dynamics are unique to this particular topology. Okay. Then you could do stochastic simulations. You could take the differential equations, add a little bit of noise to it, and what you see is that yes, in time you see cells switching back and forth. So here is a region in which, so green is this is A, this is B, this is C. So in this region, green is high, red and blue are low, so cells are in state C. Here cells are in state B, here cells are in state A. So you could actually see that these cells could switch back and forth given sufficient amount of noise which is present in the system. The other question we had was that if you now look at this system, toggle triad. So toggle switch is simple, right? Toggle switch simple in the sense that if A goes high, then B goes low. And because B is low, A will stay high, right? What happens here? A goes high, B goes low, C goes high, but A and C can't be high together because A and C are also inhibiting each other. Absolutely, yes. And
should I consider it as two negative cycles or totally a single positive cycle?
it is not, I think it's somewhere in between. It's probably not either of them. It's some combination between. That's my first guess, but I can discuss.
As soon as you have a positive cycle, then you have multi-stability,
Yes.
bistability.
Yes.
You
Yes.
are getting tri-stability.
Yes.
So can we understand this in terms of how many positive and negative cycles we have?
We can do that. In fact, now that we have those other control networks where we have replaced one or more with them, we could look at similarly the frequency of bi and tri-stability in that, and then try to see if this could be explained purely based on topology.
When you had that histogram, so the histogram that you were showing, right, this one. So essentially what you're saying is that you looked at the parameter space in an exhaustive fashion, or largely exhaustive fashion, and the histograms is showing how often you see
Yes.
these as the steady states
Yes.
of the system.
Yes. Yes.
Thanks.
No. No time delay.
Okay.
Why I'm saying, because I mean, if you don't consider the, it looks similar to a repressolator.
Yes, yes, you are absolutely right.
So, but there is like two repressolators in anti-mode.
So yeah, absolutely. So when we simulated this, we saw, I think, 40% of the cases it was giving oscillations.
Yeah, yeah.
Then we added this, it came down to 20, 10, 1. So the oscillations die down very, very quickly the moment you add the other cycle.
So is there anyone, like, I don't know, as in analytically, can this be kind of
It can be
justified?
solved. I mean, we have not done it,
Oh, okay.
but this is absolutely right. I mean, this is an open question, that how does it change? And also what Arijit was asking, how do you treat this as a combination of cycles, or coupled cycles or decoupled cycles and so on?
And also one thing that the histogram that
Yes.
shown, so that's previous. Yeah. Can anyway this be related to a XOR gate? I mean, if all the proteins kind of, let's say, activate some other protein maybe. So you are seeing, like, when all the, like, you are not seeing, I mean, when you are not seeing proteins which are like all are active, and
Yes, yes, that
yeah,
last one
all-
is not,
yeah,
they're not
and
seen.
if all are active, that is also like the chances are very low,
Very
and
low.
all are inactive, that are chances
Yes.
also very low.
Yes.
So is this like similar to what an XOR might?
No, I don't know that, so maybe I'll need to understand that.
Yeah, I mean, let's say these, I don't know what these proteins are, but let's say there are 3 proteins A, B, C.
A, B, C, yeah, let's go with
Yeah,
A, B, C,
so,
right.
I mean, so basically, if you have both A, B, C, both as active, and it gives a low output.
Right. Oh, XOR gate, you mean?
Yeah, XOR
okay,
gate.
okay,
Yeah,
okay,
yeah,
right,
yeah.
right.
So, and if both are low, and if all are low or if all are high,
Right,
then you
then
get
you
a low.
don't get,
I mean,
yeah.
So that is what you are seeing actually.
Yes, yes. In this
sort
case,
of.
you could simplify it to that Boolean logic being followed at all nodes.
Okay, thank you.
So then we ask this question, then, you know, if the system is so frustrated that it's hard to get a steady state out of this because they both also cannot be high together, then how do you get this output, right? So then is when we started looking more carefully at the parameters, because the topology here seems to be insufficient to tell which output you'll get. So you need to look at the parameters. So we basically, you know, non-dimensionalize the system. We identified this parameter, which is basically telling you the link strength, right? So again, if you if you go back to the Hill's function, so G by K, the G is the production, K is the degradation, H naught is the threshold. So H naught by G by K is basically a measure of, you know, how much normalized levels would you need of a protein to be able to inhibit the other. So that's non-dimensional. Lambda is basically just a fold change, and N is the non-linearity of the system. Okay, N and lambda have no units. So when you take this, you take all the parameters in which you found A was high, B and C were low, and you calculate the values of those cases in each parameter set, and ask, you know, one scenario in which this could happen is that you have A high, B, C low, is that when A is inhibiting C much more strongly and A is inhibiting B much more strongly than B is inhibiting A or than C is inhibiting A. If this condition is satisfied, then B and C would go very low, and because they will be low, they will not be able to inhibit each other as strongly because they are perhaps lower than their own threshold. So this is one scenario in which this frustration could potentially be resolved. So that was our hypothesis. So we took every single parameter set and asked that in every single parameter set that gave us monostable this state as the output, when is this condition satisfied versus when is this condition not satisfied. Okay? So the answer we got is the following. Indeed, in 90% of the cases, this condition was true, and 90% of the cases this condition was true. And as you can see, between B and C, there was no major asymmetry. B and C were inhibiting each other to relatively equal levels. Whether that was strong or weak is a separate discussion, but there was no strong bias that was happening in this case, right? And we also saw that those 90% and that 90% was largely overlapping with each other. So basically, you need A inhibiting B being stronger than this, and A inhibiting C being stronger than this to be able to get that. What happens in remaining 10% of the cases, we have not yet analyzed. Okay. Now let's take bistable combination where C is low in both of them, but A and B switch. Now let's see what is a possibility. So if C is low and A and B are switching, one possibility is that A and B cannot, there cannot be a large asymmetry in this because then you will not be able to switch. But C, the poor guy, is being low in both the cases. So this has to be strong and this has to be strong as compared to this. You could only compare in pairwise. You can't compare across pairs, right? Please. So lambda, so the function, lambda is a fold change. So the function that is written is something like, you know, so if you put lambda equals to 1, then this function becomes 1. So there is no regulation. A is not impacting B. If lambda is much greater than 1, then the activation part is stronger. If lambda is less than 1, then the inhibition part is stronger. So it's just a way to compress parameters, that's all. So you could have both activation and inhibition in the same function. Lambda 1, no regulation, lambda greater than 1 activation, lambda less than 1 inhibition. Okay. And indeed, this is what we saw. A and B, there was not a major asymmetry, but C was being inhibited much more strongly by A and B to allow for that combination to happen. So basically, again, you know, you don't always get all the information from the topology. You would have to look at parametric combinations in some cases to be able to identify how you see different possible states out of it. Now, of course, this has been shown, the 3 states that we talked about, they have been well reported, but also more recently, people have shown that these hybrid states can exist stably over multiple generations in vivo. So, you know, whatever our model predicted that you could have 6 states are all have been reported in this case. We looked further more detail into, you know, the RNA sequencing data, where you have in the same experiment naive cells which means the progenitor cells, TH1 cells and TH2 cells. X and Y axis are some ensemble measure of gene expression values corresponding to these states. So what you see is that when you look at TH1 cells, TH1 signal is much stronger than TH2, and in TH2 cells, the TH2 signal is much stronger than TH1, as expected, right? You could also look at another experiment which was done that had time course data, and you could see that, you know, in both TH1 and TH2, while they started from relatively equal levels, clearly the divergence started to happen soon enough, and you could see that only increasing as a function of time. There was thankfully one experimental data set where the same cells were pushed to become TH1, TH2 and TH17. And again, you could see that in TH1 cells, the TH2 and TH17 were lower. In TH2 cells, the TH1 and 17 was lower, and in TH17 cells, everything else was lower except TH17. So basically, cells are indeed making decisions, indeed the way that our model predicts. If you have this longitudinal kind of data from the same experiments, you could make that claim. Okay, last experiment, last network before we close. 4 molecules inhibiting each other. What do you think is going to happen? How many of you think, so there are now 16 possible states, 2 to the power 4, right? One state is all low, one state is all high. 4 states are single positive, 4 states are triple positive, and 6 states are double positive, right? It's a completely symmetric system again, and there is an example for this in T cell differentiation. How many of you think you would get single positive states? You would get single positive states. Some single. What percentage? Approximately, give me a ballpark number. So in the previous one, I think single positive was around 55% and double positive was around 40, if I remember correctly, somewhere of that. The difference was not very high. So here you are saying 30. Okay. Anyone else? Sorry? 25 to 30%. All single positive put together. And what will be the remaining 70%? Double positive, triple positive, both. Okay. Any more answers for frequency of single positive? How many of you think it would be greater than, say, 50%? Greater than 50%, okay. 50 to 30. We're using the same strategy to simulate. Sample large parameter sets, simulate. 30 to 10, or 10 to 30. Okay. 1 to 10. Less than 1%. The answer is 1%. You don't get single positive states at all when you look at this network. What you get is all double positive states, too high, too low. Why? We don't know, but this is what the results you get. So the way we are interpreting this is that if a cell has more than 3 choices to make, it cannot take that decision in a single step. It first eliminates 2 choices, as if it was playing Who Wants to Be a Millionaire or Kaun Banega Crorepati. Eliminate 2 choices and then go to the next step. And probably that is why we do not see more than 2 or 3 branches coming out from the same node. So the next question we asked is, okay, maybe this network cannot give us. Let's look at any other 4 node network. Okay? So you take this network, and let's say if you represent this network as a matrix, as a 4 cross 4 matrix, and each element of that matrix, row is the source, column is the target, each element of that matrix can be +1, -1 or 0, activation, inhibition or no regulation. You could have how many possible networks? 3 to the power 16 networks, -1, of course. None of those networks give single positive states as the most dominant one. So it's not just this network. It looks like none of the 4 node combinations is allowing cells to make that decision in a single step. Then again, we did similar analysis. When in whatever limited number of cases you do get single positive, what is the condition? So the condition is, of course, very straightforward, that D is inhibiting others much more strongly as compared to vice versa. D inhibits B much more strongly, D inhibits A much more strongly, D inhibits C much more strongly. What if you have this combination? D is high, A and B are switching. So again, you can figure out the scenarios that A is switching, so A and B cannot be much high because they're switching. C is constantly low, so C is always inhibited more strongly, and similarly you can make up other combinations and get these kinds of conditions. You could do stochastic switching, stochastic simulations, and you can see that only 2 elements are switching at a time. So there are 2 elements which remain constantly low or constantly high, and 2 which switch from high to low or low to high. So basically what we are saying is that in this kind of a structure, you first choose any of the 6 double positive states and then eventually go to one of these 4 positive states. The last question we asked was, let's look at 6 node, 8 node networks. What happens there? 6 node networks, the most frequent ones are triple positive. So it follows the pattern. Toggle switch, most frequent is single positive. Tetrahedron, most frequent is double positive. Whatever this is called, most frequent is triple positive. 8 of them, most frequent is 4 positives. So if you have to, a cell has to choose from here, if such a structure exists. I'm not saying this structure exists, but if such a network exists, it will have to cut down from 8 to 4, 4 to 2, 2 to 1. What happens if you look at odd cases? So we have already looked at toggle triad, and there we saw single positive and double positive were comparable. When we look at the 5 node one, again it's the 2 and 3 node which is most. When you look at 7, it's 3 and 4. So again, this pattern is also constantly maintained. So none of the networks beyond a toggle triad gives you single positive states as the most dominant states, and that could possibly explain why you see that this decision making cannot be done in single step. So you start from, and so far, at least I am very surprised by this, toggle triad has not been synthetically constructed. 25 years since toggle switch was constructed synthetically. It's just one more node that has to be added, and the synthetic biology colleagues I have spoken to also do not know of any such effort which was successful. It was done, but perhaps it was not successful. So clearly, you start from a single simple network and just add a little bit more complexity in the dynamics changes, add one more variable, and you see a fundamentally different dynamics, which again has a reason. I'm not saying we understand the reason, which matches with what is known experimentally. Why 3 is the limit? I have no answers. That is something which we are still thinking about. Okay, thank you very much for your attention, and I'll continue tomorrow.
Thanks, Mohit. Since there were quite a few questions during the talk, we'll now break for coffee. But please feel free to catch Mohit during the coffee break if you have further questions. So coffee again is just outside, and we should be back at 4 for the final lecture today by Professor Somnath Sinha.